Okay, so I'll share my screen and we can start the session. Okay, so I'll start today's class. Okay, so good evening. Today is the 8th live session of this sequence of live sessions that we do for this course Biochemistry. So this course is taken by Professor K. Subramaniam, and basically what we do in these live sessions is that we discuss some basic concepts behind some questions, and we try to discuss all the patterns of the questions and how to solve those kind of questions. We talk about the basic concepts so that it's easier for us to solve different questions based on the same concepts or applications. And my name is Aishi and I'm from IIT Bombay. So I'll move on to the first question for today that we'll discuss. Okay. So for amino acid sequencing of a protein or a polypeptide, Sanger's method and Edmonds process were the earliest biochemical methods, both of which involved separating the number 1 dash terminal amino acid of the protein. The reagents used in Sanger's method included 2 dash, while Edmonds degradation used 3 dash. So there are all these 5 options that there are, and we have to figure out the correct answer for this question. Okay, so we'll discuss basic concepts behind the Sanger's method and Edmonds process and all of that, and we'll discuss the answer also, but it would be you can take a minute to think about it and we can then move ahead with the discussion. So I'll wait for a few minutes and then I'll move ahead with the discussion. Okay, so the correct answer for this will be number 1 will be amino acid. So both of these processes involve separating the amino terminal amino acids of the protein, and the reagents used in Sanger's method will include FDNB and HCl, while Edmonds degradation will use phenyl isothiocyanate and CH3COH. Now we'll discuss about these processes in brief. So we know basically that our life is maintained by a molecular network system. So this is basically a molecular network systems, and this entire thing. And if we zoom into each of the molecular network systems, we can see the different pathways, the different cascades and everything. Okay, so proteins play key roles in a living system, and then different types of proteins with different types of functions are there. So there are 3 examples of protein functions: catalysis, where almost all chemical reactions in a living cell are catalyzed by protein enzymes; transport, where some proteins transports various substances such as oxygen, ions and so on; and information transfer like hormones, etc. So if we have to look at the examples, like alcohol dehydrogenase, it oxidizes alcohols to aldehydes or ketones. Then hemoglobin carries oxygen. Then insulin controls the amount of sugar in the blood. So these are the different types of protein functions belonging to different proteins. So now the estimated functional roles by percentage of proteins of the proteome in a complex organism. Okay, so if you can see that this is a complex organism. 2% of the proteins might be involved in transport. 22% might be involved in metabolism, 8% in signal transduction, 6% in energy, 3% in protein synthesis, 5% in protein destination and storage, then 13% in disease and defense, 6% in growth and division, 15% in transcription, 8% in cell structure, 10% in secondary metabolism, 2% in intercellular traffic. So if we can categorize everything on a percentage basis, these are the different estimated functional roles of the proteins, of different proteins, and the main functions that they are involved in the protein of a complex organism. Okay, so what are the protein functions, major protein functions? Transport, regulatory, motor, fibrous protein, enzyme and immunoglobulin. Right? This is the basic structure of an amino acid. Okay, so this is the amino acid glycine. No, sorry, this is the basic structure of an amino acid. If there is an H hydrogen over here also, it becomes glycine. The simplest amino acid structure. Okay, so we have an amino group, we have a carboxy terminal and carboxy group, and we have a side chain. So basically all the amino acids have these three features constant, common. The side chain is what gives the amino acid its specific properties. Okay, so that is dependent on the side chain. So you see you have L-alanine and D-alanine. Okay, that's so if you can see, this is the structure of L-alanine, but H is, they are basically non-superimposable mirror images of each other. If you can see the different forms of the structure L-alanine and D-alanine. Now there are 20 amino acids that we have talked about: lysine, alanine, valine, isoleucine, leucine, proline, methionine, phenylalanine, tryptophan, asparagine, glutamine, serine, threonine, tyrosine, cysteine, aspartic acid, glutamic acid, lysine, arginine, and histidine. So what do the white labeled amino acids indicate? They are the hydrophobic amino acids like lysine, alanine, valine, isoleucine, leucine, proline, methionine, phenylalanine and tryptophan are hydrophobic amino acids. Whereas the green ones are hydrophilic like asparagine, glutamine, serine, threonine, tyrosine, cysteine. These are hydrophilic amino acids. Then we have the acidic amino acids, which are the aspartic acid and glutamic acid, the red amino acids, and then we have the blue are the basic amino acids, lysine, arginine, histidine, etc. Lysine, arginine, histidine. So what are alpha amino acids? So all proteins are composed of amino acids. Amino acids are the building blocks of proteins. Now there are 20 common amino acids that exist. Now all amino acids are primary amino acids except for proline. Proline is a secondary imine basically. So primary amino group is attached to the alpha carbon of the carboxyl group. So except for glycine, all other amino acids have at least one chiral center. All chiral amino acids belong to the L amino acid group. So since glycine has two edge bonds, two edge groups attached to attached to the C alpha carbon, so that's why glycine does not have any chiral centers. So if you see, these are the structures of L-glyceraldehyde, D-glyceraldehyde, L-alanine and D-alanine. You will figure out the difference. Now what do these amino acids vary in? They vary in size, structure, electric charge and even solubility of water. So if we try to measure or compare the properties of amino acids, there are a huge bunch of things that we can talk about. So classification of amino acids can be done by the polarity of the side chains, hydrophobic, that is water fearing, non-polar side chains, hydrophilic, which is water loving, polar neutral side chains, and we have positively charged, negatively charged based on the charge. And then we have aromatic amino acids as well. So these are the non-polar aliphatic R groups: glycine, alanine, proline, valine, leucine, isoleucine, methionine. These are non-polar and they have an aliphatic R group. Then we have the aromatic R groups, which if you can see, there is an aromatic. There's basically a group over here, which makes them aromatic R groups. So we have phenylalanine, tyrosine, and tryptophan, and these should belong to the aromatic R groups section of amino acids. Then we have the polar uncharged R group. So there are some amino acids which are polar, but they are not really charged. They do not carry any particular charge, positive or negative. So we have the polar uncharged R groups, like serine, threonine, cysteine, asparagine, glutamine, all of these. Then we have the positively charged R groups, like lysine, arginine, and histidine. They have a positive charge. And because of the R group difference in these two amino acids, they have different from the other amino acids. They have a NH2 NH3 group in their side chain, which makes it really positively charged. And histidine is not like that. Histidine is not like that. In case of histidine, histidine is also a positive charged amino acid. Basically, the side chain, the charge of the side chain will determine basically the charge of your amino acid. Next, we have the negatively charged R groups. For this, we have the aspartate and the glutamate, the acidic amino acids. So we have an extra COO functional group in the side chain, which makes these amino acids negatively charged. So, for example, if you want to form a disulfide linkages by cysteine and cysteine, we can bring them together with some reaction, and we can get glycine basically. You see the S bonds have attached, and that is glycine. Sulfur have attached. So there are other non-standard amino acids as well. There is the 4-hydroxyproline, then 5-hydroxylysine, 6N-methyl and 6N-methyl-lysine, 4-hydroxyproline, 5-hydroxylysine, and gamma-carboxyglutamate. Sorry. And obviously there is cysteine. So apart from the standard amino acids, we also have some non-standard amino acids, which makes the list quite bigger. So these are some of the non-standard amino acids. We can have phosphoserine, phosphate group attached to serine, phosphothreonine, a phosphate group attached to threonine, phosphotyrosine, and N-methyl-arginine, acetyl-lysine, glutamate, methyl ester, adenylyl tyrosine, all of these. So we also have amino acids ornithine and citrulline, which have other significant relevance in the system. Now, if you can see over here, there is the absorbance curves of tyrosine and tryptophan. You can see at which point they absorb the most. So we also talked about Beer-Lambert's law in one of the previous classes. Basically, there will be a beam of light incident. Light will be labeled as I naught, and there will be a sample cuvette with a particular path length. Okay, like sample cuvette holding the sample in a particular path length, and the intensity of the light will be measured by a detector. That is what basic principle of Beer-Lambert's law is. We talk a little bit about the titration curve of amino acids for neutral, acidic and basic side chains. This is the titration curve of glycine. You can see the different pK values and the PI. PI is basically the isoelectric point, which denotes the point at which lysine doesn't carry any particular charge. So that is the PI or electric point. Then for glutamate we have a different titration curve, obviously, which is really expected. For histidine, it is also already in the part of doing something big. So you can see the OH equivalents are on the x-axis, whereas the pH is on the y-axis. And this is the titration curve of histidine. So we have already talked about the different categorizations where how we can divide amino acids. Now coming to peptide bond. When two amino acids are coming together, two or more, there is formation of peptide bond between the two amino acids. There is a CONH bond formed. There can be different types of peptides: dipeptides, tripeptides, oligopeptides, polypeptides, and ultimately proteins. Give me a moment. There's some network issue. 1 second. Not able to change the slides. One second. Give me a moment. I'll check. Yeah. So I'm back. So these are different types of peptides. It can be dipeptide, tripeptide, tetrapeptide, oligopeptide, etc. Okay, so if we check systems for this is a peptide bond. There will be an N-terminal, amino terminal, C-terminal, carboxy terminal. This is the peptide bond. Okay, these are the two amino acids that have been linked together. So if you want to talk about the nomenclature, you just have to replace the INE by YL, and we have to keep the last INE. So that will, if we have a tripeptide alanine glycine arginine, we will say alanyl glycyl arginine. We already talked about the 4 different levels of structures of proteins. So there is a primary structure, secondary structure, tertiary structure, quaternary structure. So we have talked about the primary structure, which is basically nothing but the amino acid sequence. The sequence of the amino acids, which gives rise to secondary structure. The main forms of secondary structure can be alpha helix or beta sheets. There can be other types of structures as well, such as turns, loops, and all of that. But alpha and beta helix are the major secondary structures when you're talking about any kind of protein component. Then that gives rise to the tertiary structure, which is basically a 3D representation of the proteins. Okay, so the structure will fold upon the protein structure will fold upon except by means of various bonds and attractions. Various types of attraction and some disulfide bonds as well. That will give rise to tertiary structure, basically a 3D representation. And then if the protein consists of various subunits and everything, then the assembled subunits will come together and will give rise to the quaternary structure. That is how the protein structure for different levels can be described. This is from a table from Leninger, the molecular data on some proteins basically. So different molecular weights, number of residues, number of polypeptide chains, all of these are mentioned. So there are conjugated proteins as well, proteins that are conjugated with some other component. For example, lipoproteins, lipids are conjugated. Lipids are the prosthetic group. Okay, for glycoproteins, the conjugate prosthetic group is carbohydrate. For phosphoproteins, the conjugate group is phosphate groups. For hemoprotein, it's heme. For flavoproteins, it's flavin nucleotides. All of these. Then from metalloproteins, you have the different types of metals: iron, zinc, calcium, molybdenum, copper. Different metals which act as prosthetic groups. Now, peptide and protein charge. So, if a peptide sequence is given to you, how do you figure out basically the protein charge of the peptide when they come together? So, basically, how do you figure out the isoelectric point of that peptide? So, what do you do in that case? We have different groups, right? COH and NH. We take the COH and NH group. The pKa values, and from that we try to figure out the pi values. Okay, so that is the basic way to go ahead with the peptide and protein charge. We think about the net charge at different pHs if we know the pKa, and then we move from there. Now, how do we do protein separation or purification? So, in general, proteins will contain various residues, and then we need to fold into tertiary structure. Okay, so proteins can be separated on the basis of differences in their size and composition. So, they must be also pure to analyze. Like further, if you want to move ahead with the protein, you have to analyze that it's pure and its structure and function is proper. So what are the factors that we need to control? We need to avoid denaturation or chemical degradation by pH, presence of enzymes, temperature, reactive thiol groups, etc., exposure to air, water, etc. And what are the different methods of separation or purification? Can be solubility, where different salts, solvents, and pH temperature, all of these are equal factors. They can be chromatography, like ion exchange, gel filtration, and affinity chromatography. These are very popular methods of separating or purifying proteins. Or electrophoresis. So electrophoresis can be used to separate proteins based on their molecular weight. So if you see this in some kind of column chromatography, we have add the mobile phase in the reservoir, and then the protein sample is in the mobile phase, and we have a solid porous matrix stationary phase and a porous support. So by then the column will go and the proteins might be able to separate based on their different factors, the different criteria that we needing to use separate the proteins. Okay. For example, ion exchange chromatography. If we look, the deep blue proteins have large net positive charge. The light blue ones have net positive charge. The orange ones have net negative charge, whereas the red ones have large net negative charge. In the stationary phase, we have polymer beads with negatively charged functional groups. Now the protein mixture is added to the column containing the cation exchanges. Proteins will move through the column at rates determined by the net charge and the pH being used. So with cation exchanges, what happens? Proteins with the more negative net charge move faster, elute faster. Okay. So what happens if the polymer beads here are already negatively charged, and then the solution that needs to be separated, the components of the solution that need the solution is poured. Now the proteins which are negatively charged in the system will get repelled by the reagents and they'll directly elute. They'll elute faster. Okay. So protein mixture is added to the column containing. Okay, so proteins with a more negative net charge will not really interact or get attracted to the negatively charged reagents. They'll be repelled on the other hand. So they'll elute faster. They'll elute earlier. Whereas proteins with net positive charge will interact with the stationary reagents, stationary phase, and they'll elute later. This for size extension chromatography or gel filtration or gel permeation chromatography. The concept or principle is a little different. So here we don't separate based on charge, we separate based on their molecular size, basically size. So what happens? The beads here are porous polymer beads, and the protein mixture is added to the column containing this cross-linking polymer. Now, protein molecules separate by size. Molecules will pass more freely, appearing in the earlier fractions. So, if there are huge molecules, larger molecules, they'll not go into the pores of these beads. The beads will not incorporate these huge molecules, huge proteins. So they'll be ousted by the porous beads. So they'll not really go into the bead. They'll not have any kind of that kind of friction. They'll basically elute faster and way earlier. Whereas the small beads will go into the pores, and then that will be a different mode of friction available. So the small beads will elute later. Then we have affinity chromatography. Here the proteins are separated based on their affinity for a particular something. So this is a column. The protein mixture is added to the column containing a polymer bound ligand specific for a protein of interest. So suppose we have a protein of interest in our mixture of proteins. Okay, and now we want to separate only that protein of interest. So, and we know that there is another compound which has an affinity or ligand. It has an affinity for a protein of interest. So we will basically attach a ligand to the beads and then we'll allow our protein mixture to pass through. So whatever happens, what happens is that when the protein mixture is being passed through, the protein of interest which will have a specific interaction with the ligand will get attached to it. The rest everything will dilute faster. Any questions till now? Okay. So this is a purification table for a hypothetical enzyme. You have seen through cellular extract how people basically extract the different kinds of proteins. If we talk about gel electrophoresis, we have talked about that in detail in one of the previous classes. We have talked about the role of SGS. It basically imparts a uniform negative charge on the protein. This is a setup. This is how we load samples. We know the duration of migration. So basically proteins get separated on the basis of the molecular size only, which is why we have markers. Markers are basically like ladders commercially purchased that have been kept in lab to basically use it in STS plate gel. Okay. So if you know the molecular weight, it's a ladder for example, and we with different bands corresponding to different molecular weights. Now if you have a protein of interest, which is an unknown protein, and you want to find the molecular weight, we can find the molecular weight by making a standard curve. Okay, so we can plot the distance traveled by each of these proteins from the starting point. Okay, and we can make a standard curve using that, and then we can use an unknown protein basically and to extrapolate the data of the standard curve. So this is our standard graph. These are different proteins. We know the molecular weight of the proteins, and we know the relative migration from the well where we have loaded the sample. Now this is our unknown protein. We don't know the molecular weight of the protein which we need to find out, but we know the relative migration of that unknown protein. So that is what we plot over here. Then we drop a perpendicular on our standard curve, and then we draw another perpendicular on the y-axis, and from here we can find out the tentative predicted molecular weight of the protein, unknown protein. This is another way of basically this is also from Leninger book. So in this case what happens is an ampholyte solution is incorporated into the gel. Now what is ampholyte? So ampholytes are nothing. Basically amphoteric compound is a molecular ion that can react both as an acid and a base. So what exactly this can mean depends on which definitions of acids and bases are being used. So one type of amphoteric species are amphoprotic molecules which can either donate or accept a proton. Now if you ask me what do you mean, ampholytes are basically compounds that when dissolved in water, which is itself an amphoteric compound, can act either as an acid or a base. Okay, so that is the primary role of ampholites. So what happens here is basically an amine, as we know that amino acids are crystalline solids with a melting point higher than those of organic molecules of similar size like amines, carboxylic acids, which are not crystalline solids. And this suggested that amino acids are dipolar ions in which the COH group occurs in its dissociated form and the NH2 group is protonated like CO minus and NH2 plus. Okay. So the strong electrostatic forces between the positive and the negatively charged neighboring molecules can explain their high melting point. Furthermore, their dipolar nature can also explain the good solubility of amino acids in water. Okay, for example, that is the basically the meaning of ampholites. So here in this case, if you see an ampholite solution is incorporated into a gel and which has a decreasing pH from pH 9 to pH 3. So there is a stable pH gradient is established in the gel after application of an electric field. Now from above, a protein solution is added and electric field is reapplied. So after staining, proteins are shown to be distributed along pH gradient according to their pI values. So what happens, the proteins are applied over here, the proteins will start moving down, correct? Depending on their charge basically, because if that's a negative protein, that will probably travel to a greater extent. So when will the protein stop traveling? That is when they have reached their isoelectric point in the pH gradient. Once they have reached the isoelectric point in the pH gradient, they will stop. Okay, so this is how we can find out the isoelectric points of some proteins. Now there is a way of doing two dimensions of separation. So the first dimension can be isoelectric focusing, which we have discussed right now. Decreasing pH, samples are added and the proteins will stop at the particular point where their isoelectric point is around. Okay, then the isoelectric, so now the proteins have been separated on one dimension. Now further separation can be done based on STS page. So now that the isoelectric focusing gel is placed on the STS polyacrylamide page, and then the second dimension gel separation of gel running STS page gel electrophoresis occurs where proteins are separated according to their molecular weight. So this side is decreasing pi, this side is decreasing molecular weight. Now we come to protein sequencing. Now there are different types of protein sequencing methods. There is a traditional chemical method which is the Sanger's method, the genetic method, and obviously there is proteomics. So protein chemists basically follow a basic strategy when they attempt to determine the sequence of most proteins. Okay, so in order to know which amino acids and how many of each amino acid there are in a polypeptide, we must break the peptide bonds, and this can be accomplished with very strong acids like 6-normal HCl or strong bases or Y ions exhaustive enzymatic digestion. So by performing an acid hydrolysis or base hydrolysis experiment, you obtain a minimum length for the polypeptide. So now the hydrolyzed amino acids are then grouped into different groups according to their pi or isoelectric point values, and each amino acid are isolated by HPLC with pH gradient buffer and determined by reacting with hydrate. So the concentration of individual amino acids are normalized to integral number, and by comparing with the molecular weight, the actual number of the individual amino acids in that particular protein can be determined. Okay. So what happens in hydrolysis? A polypeptide can be hydrolyzed by refluxing with 6 molar hydrochloric acid for 24 hours. Now the individual amino acids can be separated from each other using cation exchange reagent. So an acidic solution of the amino acids is passed through the cation exchange column. Strength of the adsorption varies with the viscosity of each amino acid. The most basic are held most strongly. Now we can watch the column with a sequence of buffered solutions which will cause the amino acids to move through it at different rates. So in the original method, apparently the column element is treated with ninhydrin, a dye used for detecting and quantifying each amino acid as it comes off the column. So that is the basically job of ninhydrin we previously used to do. So what is Sanger's N-terminal analysis? The N-terminal end of the polypeptide is labeled with 2,4-dinitrofluorobenzene and the polypeptide is then hydrolyzed. And the labeled N-terminal amino acid is separated from the mixture and then identified. So that is basically the Sanger N-terminal analysis method. Then there is a C-terminal analysis method. An enzymes called carboxypeptidases hydrolyze the C-terminal amino acid respectively. Now the enzyme continues to release each new exposed C-terminal amino acid as the peptide is hydrolyzed, and it is necessary to monitor the release of C-terminal amino acids as a function of time, basically to identify and verify them. We have talked about the primary structure of proteins, the sequence of amino acids. Now there's Edmond degradation, which basically involves sequential cleavage and identification of N-terminal amino acids. So Edmond degradation works well for the polypeptide sequence analysis up to approximately 60 amino acid residues. So the N-terminal of the polypeptide reacts with phenyl isothiocyanate, and the resulting phenyl thiocarbamyl derivative is relieved from the peptide chain. So the unstable product rearranges to a stable phenyl hydantoin. It is purified by HPLC and identified by comparison PATA standards. So an automated amino acid sequencing machine uses the Edmond degradation high performance liquid chromatography. So core Edmond degradation cycle beginning with a picomolar of polypeptide can be completed in approximately 30 minutes. So each cycle results in identification of the next amino acid residue in the peptide. So this is how it works. The disulfide bonds are broken by oxidation of by performing acid or reduction by DDT basically. So from there we got carboxymethylated cysteine residues. So the Sanger and Edmond methods of analysis apply to short polypeptide sequences. So for large proteins and polypeptides, the sample is subjected to partial hydrolysis and with dilute acid to give an random assortment of sort of polypeptides which were then analyzed. So the smaller polypeptides are sequenced. The rounds of overlap among the stem allow the entire polypeptide to be sequenced. Now, larger polypeptides can also be cleaved into smaller sequences using specific reagents and enzymes. So, the use of agents gives more predictable fragments which can again be overslapped to obtain the sequence of the entire polypeptide. Uh, Cyanogen bromide here, which cleaves red bonds only in the C-terminal side of the methionine residues. So mass spec can be used to determine polypeptides and protein sequences. Okay. This table shows specifics of common methods for fragmenting polypeptide chains. Now the initial cleavage is generally made as specific as possible in order to generate large peptide fragments, and it is easy to arrange fewer fragments. These fragments can be in any position relatively to one another. After treatment of the original polypeptide that generates fragments whose sequences extend across the initial cleavage points as well. So locate the disulfide bonds. So no primary structure analysis of a cysteine containing protein can be regarded as complete before the presence and location of disulfide bond has been established. And we can reconstruct the original protein. So from the overlapping peptides and information gained from the original protein, a unique sequence for the protein or polypeptide of interest can be worked out. Okay, so if you see this, there is 2,4-dinitrophenyl derivative of polypeptides FDNB, and then 6 molar HCl is needed to form 2,4-dinitrophenyl derivative of amino terminal residue, and obviously some free amino acids. Okay, and then there is also need of phenyl isothiocyanate, and with the OH group, basically there is a interchange of these things. So so this is a phenyl isothiocyanate, and then we add it to the PTC adduct, and then anilinothioazolinone derivative of amino acid is formed, which ultimately gives rise to the phenyl thiohydrantoin derivative of amino acid residue. So we have the procedure hydrolyze separate amino acids. So it can react with FDNB, hydrolyze separate amino acids. It can be used on sulfide bonds and also unnati. What happens is that here, so if you see that here the procedure is that we hydrolyze and separate the amino acids. This is polypeptide, it will react with FDNB, hydrolyze, separate the amino acids, and the disulfide bonds will be reduced if they are present. Now we then cleave with trypsin. Trypsin will cleave at the C-terminal end of arginine and lysine of the basic amino acids, and the fragments will be separated and sequenced by Edmond degradation. And then we can cleave with cyanogen bromide, separate the fragments and sequence by Edmond degradation again. And then we can establish the sequence basically. That is how this happens. This is amino acid sequence and other mRNA sequences that specify the same amino acid sequence. Now there is something called ladder sequencing. Basically involves analyzing a polypeptide digest by mass spec, wherein each polypeptide in the digest differs by one amino acid in length. The difference in mass between each adjacent peak indicates the amino acid that occupies the position in the sequence. So mass spectra of polypeptide fragments from a protein can be compared with databases of known polypeptide sequences, this leading to an identification of the protein or a part of its sequence by matching. We have also talked about the acid-base behavior of amino acids. These amino acids basically exist as deuteran. It is a dipolar ion having both formal positive and formal negative charge. So the overall charge will be neutral. So amino acids are amphoteric. They can react as either an acid or a base. Ammonium ions act as an acid, carboxylate as a base. And the isoelectric point where the pH at which the amino acid exists largely in a neutral and deuteranic form which is basically influenced by the nature of side chain. So we talked about electrophoresis, basically the separation of polar compounds based on their mobility through solid support. So if you talk about trypsin and chymotrypsin, they are basically endopeptidases. So there is a carboxypeptidase where it leaves the amide bond of the C-terminal amino acid. Exopeptidase. Now the C-terminal amino acid is identified by treating with peptide with carboxypeptidase and analyzing by liquid chromatography. You can also talk about end labeling, which is basically the peptides is first treated with 1-fluoro 2,4-dinitrobenzene, which selectively reacts with the N-terminal amino group. The peptide is then hydrolyzed to other amino acids, and the N-terminal amino acid identified is at it as N2,4-dinitrophenyl derivative. Now, insulin, if we talk about insulin, insulin has two peptide chains. The A chain has 21 amino acids, and the B chain has 30 amino acids, held together by two disulfide linkages. So, chemical method for the sequential cleavage and identification of the amino acids of a peptide one at a time, starting from the N terminus, is shown over here. From methyl and methyl thiohydantoin. So what happens during the Edman degradation? They have to cycle the pH to control the cleavage of the N-terminal amino acid by PTC. You have to monitor the appearance of the new N-phenyl thiohydantoin for each cycle, and it's good for peptides. Before peptides, longer peptides and proteins must be cut into smaller pieces before Edman sequencing. There are various places, polystyrene polymers are also there to give them some support. I have already talked about enzymes, which are basically proteins that catalyze biochemical reactions by bringing the reactive atoms together in the optimal geometry for the reaction. It basically lowers the activation energy by stabilizing the transition state and high energy intermediate. And many enzymes use the functional groups of the amino acid side chain to carry out the reactions. Now, what are coenzymes? Coenzymes are basically in this some reactions where there is an additional organic molecule maintenance requirement. So these are referred to as the cofactors. So the protein will not function without these coenzymes. We already talked about all these peptide bonds and the different types of amino acids. I'm not going into detail too much. We talked about the titration curves of some different amino acids. We have talked about the anionic chromatography, which is basically the charged molecules of interest are exchanged for another ion on a charged solid support, and the resins will contain negatively charged groups interacting with the positively charged molecules, which will elect from the resins by changing the pH buffer or the salt ion. For thin layer chromatography, amino acids are absorbed on a thin layer of silica gel and are separated by default separated based on the solvent migration. As in case of reverse phase high performance liquid chromatography, the amino acids are reversed on the separated on the basis of their polarity by the use of a column having a non-polar liquid immobilized on an inert matrix stationary phase. So more polar liquid reserves as the mobile phase. The amino acids are diluted in proportion to their solubility in this more polar liquid. So if you talk about the method for amino acid detection, one of them is for Edmond degradation, where phenyl isothiocyanate or the Edmond reagent will combine with the free amino acid terminus of a protein. So not only this identifies the N-terminal residue of a protein, successive reaction cycles can reveal the amino acid sequence of a peptide as well. There are other methods of protein sequence analysis as well, like amino acid sequence determined by the analysis of the gene sequence. It's possible to obtain the sequence of the protein directly produced through the translation process, but not most of the post-translational modifications. These are the peptides of biological interest and the different amino acid sequences that they have. Proteins can be classified according to their biological roles as well: catalysis, structural role, movement, defense, regulation, transport, storage, adaptations to environmental changes. We've talked about all of these. So acid hydrolysis of peptide will give a mixture of amino acids, and this mixture is separated by annexed chromatography, which depends on the differences in isoelectric point among the various amino acids. So the amino acids are detected using hydrate. Now, the acid hydrolysis of the peptide leaves all of the peptide balls. So, leaving some but not all peptides is smaller fragments, and these smaller fragments are separated, and the amino acids present in each fragment determined. So, for Sanger's method, the key reagent is this 1,4,2,4 dinitrobenzene. So in Sanger's method, the acid hydrolysis leaves all the peptide bonds, leaving a mixture of amino acids, only one of which bears the 2,4-DNP group. So for element degradation, that's a method for determining N-terminal amino acid, can be done sequentially one residue at a time on the same sample. Usually one can determine the first 20 amino acids from the N-terminus by this method. So what happens if vinyl isothiocyanate reacts with the amino hydrogen of the N-terminal amino acid? The product is phenyl thiocarbamyl derivative. So the product that forms is the thiazolone. Under the conditions of its formation, thiazolone will rearrange to form a phenyl thiohydrin twin derivative. Talked about all of these. So these are the steps towards protein sequencing. You have to prepare the protein for sequencing, determine the number of chemically different polypeptides, cleave the protein's disulfide bonds, separate and purify each segment, subunit and determine the amino acid composition for each peptide. Now, the Edmond degradation has been automated as a method to sequence proteins, and the PTH amino acid is soluble in solutes that the protein is not. This factor is used to separate the tagged amino acid from the remaining protein, following the cycle of labeling, degradation, and separation of continue. Even with the best chemistry, the reaction is about 90% efficient. After sufficient cycles, more than one amino acid is identified, making the a significant sequence determination error prone at longer reads. So if you go back to our answer, so this is the basic of Edmond degradation, where the product is a thiazolone. Under the conditions of its formation, thiazolone rearrangement will happen to a phenyl thiahydrogen derivative. Next, that was the question that we started with, and we have already discussed the question. I'll go back to the question once so that we can recapitulate whatever we learned. So for amino acid sequencing of protein or polypeptide, Sanger's method and Edmond's process or the early biochemical methods, both of which involve separating the terminal amino acids of the proteins. The reagents used in Sanger's method included FDNB and HCl, and for Edmond degradation it would include phenyl isothiazonate and CH3COOH. So that is the correct answer. So we'll move to the next question for today. Give me a second. So this is our next question for today. Anybody? Am I audible? Okay. This is our next question for today. You can try and give me the answer. I'll wait for a few minutes before I move ahead with the discussing the question. Anyone? Okay. So which of the following compositions of enzymes is true? Apoenzyme plus holoenzyme will give rise to coenzyme, or holoenzyme and coenzyme will give rise to apoenzyme, or coenzyme and apoenzyme will give rise to holoenzyme, or coenzyme plus prosthetic group will give rise to holoenzyme. We discussed a part of this question last day, but I will like to discuss it again because the discussion was not complete. Okay, so the answer will be coenzyme plus apoenzyme equal to holoenzyme. Any questions regarding the answer? So the enzyme without its non-protein moiety is termed as apoenzyme and it is inactive. Holoenzyme is an active enzyme with its non-protein component. If you can see the diagram over here, apoenzyme has the protein portion, but it is inactive on its own. Then we add the coenzyme, which is a non-protein component. It acts like an activator. And when they come together, the holoenzyme is the whole enzyme becomes active. So what are the basic roles of enzymes? Enzymes increase the reaction rates by decreasing the activation energy. There are various enzyme substrate interactions. Formation of enzyme substrate complex by lock and key or induced fit model. So if you see the graphs over here, lactose is the substrate, glucose plus lactose are the products. You see the activation energy without the enzyme is so huge, it's a huge barrier. But the activation energy with the enzyme is so less. Okay, so the catalysts are not really taken up or consumed in the reaction. They act to decrease the activation energy with the enzyme. Now, in the lock and key model of enzyme action, the active side of the enzyme has a rigid shape. So only substrates with the matching shape will be able to fit the active side of the enzyme. Substrate is a key that fits the lock of the active side. This is an older model, however, and does not really work for enzymes nowadays. This is a full concept of lock and key. So from before only, it means that from before only the active side of the enzyme is well made for the substrate, proper substrate. Enzymes have various degrees of specificity for substrates. So they may recognize and catalyze a single substrate, absolute, a group of similar substrates, or a particular type of bond. Now, what are the factors that determine enzyme activity? Environmental conditions, cofactors and coenzymes, enzyme inhibitors, etc. So, if we talk about environmental conditions, extreme temperatures are the most dangerous. High temperatures will denature or unfold the enzyme, unfold the 3D structure of the protein, and then the protein will no longer be functional. pH is also very important. All proteins or enzymes will work at a particular optimum pH. And as a substrate concentration is also very important. It's an environmental condition because it's a substrate concentration that determines the next steps. Now, what are cofactors and coenzymes? Are they different or are they the same? So, inorganic substances like zinc and iron and vitamins are sometimes needed for proper enzymatic activity. Okay? For example, iron must be present in the quaternary structure of hemoglobin in order for it to pick up oxygen. Then optimum temperature. So temperature at which the enzymatic reaction occurs the fastest. That is the optimum temperature. So different enzymes will have different optimum pH, different optimum temperature ranges. As I said, pH also affects the rate of enzyme substrate complexes. So most enzymes will have an optimum pH of around 7. But some enzymes may prefer to work or function in acidic or basic conditions like pepsin. It works in acidic conditions. Now, the rate of reaction will increase as substrate concentration increases at a constant enzyme concentration, and the maximum activity will occur when the enzyme is saturated. What does saturation mean? It means when all the sites, enzyme sites are binding, are already bound. Now in this context, if we talk about inhibitors are basically compounds or molecules which basically inhibit the action of an enzyme. They can do that by various ways. They can either compete with the main substrate, they can either overpower the main substrate, or whatever they are doing. So there are different types of inhibition. A competitive inhibitor basically will have a structure exactly similar to the substrate. So it will compete with the substrate for the active site, and it has its effect reversed by increasing substrate concentration. So as you increase substrate concentration, the effect will be reversed because it was having a competitive interaction with the inhibitor. In non-competitive inhibition, the structure is different from the substrate. It distorts the shape of the enzyme, which alters the shape of the active site. So the molecule has come and it has destroyed the shape of the enzyme. So now the enzyme is not capable of attaching, active site is not capable of attaching to substrate. It basically prevents the binding of the substrate, and it cannot have its effect reversed by adding more substrate. So what are the general properties of enzyme? Increased reaction rate, sometimes 10 to the power 6, 12 increase. They increase the reaction rate, but they do not really change the Gibbs energy, the delta G. When enzyme is present, the reaction can move forward in milder conditions. There will be reaction specificity, and it can be regulated as well. Now, non-covalent bonds and forces are maximized to bind substrates with considerable specificity. There are various bonds such as van der Waals, electrostatic hydrogen bonding, and hydrophobic interactions. Now, coenzymes, as we discussed, are smaller molecules that aid in enzyme chemistry. Enzymes can carry out acid-base reactions, transient covalent bonds, cation interactions. Enzymes cannot do oxidation reduction reactions, carbon group transfers. So, what is the prosthetic group? It basically is permanently associated with an enzyme or transiently associated. Holoenzyme is catalytically active enzyme with cofactor. Apoenzyme is enzyme without its cofactor. There are common coenzymes such as biotin is a coenzyme for the reaction carboxylation reaction. Cobalamin or B12 is a alkylation transfer reaction coenzyme. Coenzyme A is for acyl transfers, flavin is for oxidation reaction, reduction, lipoic acid is for acyl transfers, nicotinamide is for oxidation reduction, pyridoxal phosphate is for amino group transfers, tetrahydrofolate transfers for one carbon group, and thiamine pyrophosphate for aldehyde transfer. There are vitamins that are coenzyme precursors. Okay, so these are the vitamins that are basically precursors of these coenzymes. Like biotin is for biocytin, cobalamin is for balamine, folic acid is for tetrahydrofluoride, etc. Folate, etc. And these are the deficiency diseases. So if those vitamins are not present, these are the deficiency diseases that can occur. Now, there are two general ways to control enzymatic activity: by controlling the amount or availability of the enzyme, or by controlling or regulating the enzyme's catalytic activity. So each topic can be divided further into various different categories. So the enzyme amounts in a cell will depend upon the rate at which it is synthesized and the rate it is degraded. The synthesis rates can be transcriptionally or translationally controlled. Degradation rates of proteins can also be controlled. But if you think from the perspective of catalytic activity, the catalytic activity of an enzyme can be altered either positively, that is by means of increasing the activity, or negatively, that is by means of decreasing the activity through conformational alterations or structural modifications. So enzymes already encountered is like oxygen, carbon dioxide, or VPG binding to hemoglobin. So although substrate binding to the enzyme may also be modified by small molecule effectors changing its catalytic site. So protein phosphorylation of serine residues can activate or deactivate enzymes, and these are generally hormonally controlled to ensure a concerted effect on all tissues and cells. So we have learned about coenzymes, which are basically, although their enzymes can catalyze reactions, they mainly do so in association with small molecule cofactors. Cofactors may be metal ions such as zinc, etc. Organic molecules can be coenzymes such as NAD in yeast alcohol dehydrogenase. So cofactors are mostly inorganic, coenzymes are organic. So what is the difference between an enzyme and a protein? All enzymes are proteins except some RNAs, because some RNAs can also be enzymes, but not all proteins are enzymes. This is a visual representation of the lock and key model. The key is the substrate, log is the enzyme, and the hole is the active site. This is the working model of a lock and key model. This we have already talked about. This is basically lowering the accretion energy of the reaction by adding enzymes. So holoenzyme is catalytically active enzyme cofactor complex known as holoenzyme, and the enzymatically inactive protein resulting from the removal of holoenzyme's cofactor is referred to as the apoenzyme. Now what are inhibitors? Inhibitors basically alter the activity of the enzyme by combining with it in a way that increases the binding of the substrate or its turnover number. Substances that reduce an enzyme's activity in this way are known as inhibitors. There are examples such as methotrexate, which chemically resembles dihydrofolate. It binds tightly to the enzyme dihydrofolate reductase, thereby preventing it from carrying out its normal function. The reduction of dihydrofolate to tetrahydrofolate. An essential cofactor in this is acidosis of DNA precursor DTMP. Now let us talk about the specificity of enzymes. The non-covalent forces through which substrates and other molecules bind to enzymes are similar in character to the forces that detect the conformations of the proteins themselves. So both involve van der Waals, electrostatic hydrogen bonding, and hydrophobic interactions. So in general, a substrate binding site consists of an indentation or cleft on the surface of an enzyme molecule that is complementary in shape to the substrate. This is called geometric complementarity. Moreover, the amino acid residues that form the binding site are arranged to interact specifically with the substrate in an attractive manner. That is electronic complementation. Complementarity. So you see, this is an enzyme complex. It basically shows the geometric and the physical complementarity between enzymes and substrates. So these are the factors that affect enzymatic activity. Temperature and pH needs to be optimum. radiation and light, activators, end products, concentration, inhibitors, and then substrate and concentration. So what is the effect of temperature? So each enzyme is most active at a specific temperature range, which can be termed as the optimum temperature of that particular enzyme. So activity of the enzyme will progressively decrease when the temperature of the reaction is below or above the optimum temperature. However, increasing temperature also causes denaturation of the enzyme. For the shape of the bell is curved, bell shape. So the most of the enzymes of the human system have an optimum temperature within the range 34 to 40. In this temperature, optimal temperature is the temperature at which the activity of the enzyme is maximum. If we talk about effect of pH, pH change alters ionization states of the amino acid residue present in the active site. It may dissociate apoenzyme from cofactor. Drastic change will denature the enzyme protein. And it's different for different people. Optimum pH is mostly 6 to 8, but for pepsin, which is stayed in acidic environment, it can be 1 to 2. For acid phosphorus, it can be 4.5. Enzyme concentration affecting enzyme activity. So when the substrate is sufficient, rate of the reaction is proportional to the enzyme concentration. Now, products formed as a result of enzymatic reaction may accumulate, and this excess of product may lower the enzymatic reaction by occupying the active site of the enzyme. Now, if we talk about substrate concentration, the reaction is directly proportional to the substrate concentration. However, this is true only up to a certain concentration, after which the increasing concentration of substrate does not really increase the velocity of the reaction, because all the active sites will be saturated. So when the maximum velocity has been reached, all of the available enzyme has been converted to enzyme substrate complex. We talked about inhibitors and how they work. The different types of inhibitors are competitive inhibitors, which basically bind directly to the active site and hence exist in different discrete competition with the substrate. The increasing substrate levels will increase the livelihood of the enzyme colliding with the substrate instead of the inhibitor, and the maximum rate of enzyme activity Vmax can still be achieved, although it requires a higher substrate concentration. For non-competitive inhibition, it can bind to an allosteric site and do not exist in direct competition with the substrate. Increasing substrate concentrations will not affect the level of inhibition caused by the non-competitive inhibitor, and the maximum rate of enzyme activity is therefore reduced. We check the enzyme activator versus enzyme inhibitor table. So activators will bind with enzymes and increase the activity. Inhibitors will do the opposite. So these are the three conditions, basically the most important conditions affecting enzyme activity. So they can be plotted and calculated according to the time taken for the reaction to proceed. So time taken can be measured according to the product consumed, product formed, the accepted consumed. And the rate of reaction can be calculated according to the formula here given. Now, factors which can influence the rate of an enzyme catalyst reaction include temperature, pH, and substrate concentration. You see the graphs also. These are some enzymes with their cofactors. You can take a look at this table. So that was all for the previous question. We talked about apoenzyme and holoenzyme, and also some of the enzyme activities, what determines enzyme, what changes enzyme activity, what are the factors responsible. Now we'll move on to the next question for today. The assumptions taken into account while deriving the Michaelis-Menten equation. So these are the five options. The values of K1 and K minus 1 are of the same order. The rate of reaction is measured when substrate concentration is high and product concentration is low. The values of K2 and K minus 1 are of the same order. Enzyme substrate complex concentration rapidly increases to that of enzyme concentration, and the rate of formation and breakdown of enzyme substrate complex is the same. So you can think about it for 1 minute and then we'll discuss. Okay, so the answer will be the rate of the reaction is measured when substrate concentration is high and product concentration is low, and the rate of formation and breakdown of ES or enzyme substrate complex is the same. These are the assumptions that are taken to derive the Michaelis-Menten equation. So what are the factors that affect enzyme kinetics? The enzyme concentration, temperature, hydrogen ion concentration, or basically pH, substrate concentration, inhibitors, product concentration, activators, physical agents, etc. Now, enzyme concentration, the rate of enzyme catalyzed reaction is directly proportional to the concentration of the enzyme. So the plot of rate of catalysis versus enzyme concentrations is straight line. Okay. Now the temperature, basically the rate of reaction will increase with temperature. There is a bell-shaped curve. The factor by which the rate of biological reaction increases for 10 degrees Celsius increase in temperature. This is an optimum temperature, mostly at body temperature, and some enzymes will be active above body temperature, such as muscle adenylate, kinase, urease, and all of that. Now, rise or fall in enzyme activity with temperature is prominent survival feature in cold-blooded animals, and in mammals, it assumes physiological importance. Okay. So, for pH, we also get a bell-shaped curve because the enzymes will show their maximum activity and optimum pH. So, that will also give a bell-shaped curve. Now, most will show at neutral pH. Most enzymes will show maximum activity at neutral pH. And since enzymes are proteins, pH will change the effect. So, the charge state of the catalytic site and the conformation of the enzyme molecules are also very important factors that are to be considered. Now, the optimum pH of various enzymes, so for trypsin it's 7.6, and pepsin it's a very acidic environment enzyme, which is 2 to 2.5. Acid phosphatase we have, alkaline phosphatase, and all of that. Now, product concentration, accumulation, it decreases the velocity. Now, when the product is being accumulated, it is decreasing the velocity of the reaction. And in biological systems, this is prevented by quick removal of the product. Activators. Inorganic metallic cation anions can act as activators by combining with substrate AS complex, changing conformation of the active site, metal activated enzymes, metal enzymes, etc. For inhibitors, it will basically make the active site unavailable to substrate, or it will change the enzyme structure. So, what are the physical agents? Basically, light, radiation, X-rays, UV, gamma rays. These are the physical agents. Like for salivary amylase, activity can be increased by red-blue light, whereas it is decreased by the UV light. Then there is substrate concentration. The rectangular hyperbola shows the Michaelis plot. Initial velocity when little substrate is reacted. So this is a famous Michaelis-Menten equation plot. Where the x-axis shows substrate concentration and the y-axis will show initial velocity. So regions for the three phases of the curve can be interpreted. So in the first phase, the substrate concentration is very low and most of the enzyme molecules are free. So they combine with the substrate molecules. Therefore, velocity is proportional to the substrate concentration. At this state, enzymatic reaction shows first order kinetics. However, in the second phase, half of the enzyme molecules are bound to the substrate. So the velocity is not proportional to substrate concentration, and at this stage, enzymatic reaction shows mixed order kinetics. In the third phase, all the enzyme molecules are bound to the substrate. So velocity remains unchanged because free enzyme is not available through those substrate is in excess. So at this stage, the enzymatic reaction will show zero order kinetics. So then we talk about the steady state assumption. So the Michaelis-Menten equation was derived in part by making several assumptions. An important one was this concentration of substrate must be much greater than the enzyme concentration. And in the situation where the substrate concentration is much greater than the enzyme concentration and at initial velocity rates, it is assumed that the changes in the concentration of the intermediate in the substrate complex are very small over time. So this condition is termed as steady state rate and is referred to as steady state kinetics. Therefore, it follows that the rate of ES complex formation will be equal to the rate of ES breakdown. So those are some of the steady state assumptions that were taken into account for deriving the Michaelis-Menten equation. So you can see this is the equation in derivation. The rate of ES substrate in the substrate complex will be this, where ET is the total concentration of the enzyme and rate of ES breakdown to product will be this. This is steady state assumption. This equation is the basis for the final Michaelis-Menten following algebraic rearrangement and substitution of Km and Vmax terms. So this is the Michaelis-Menten equation, where V naught is the initial reaction velocity, Km is the Michaelis constant, and Vmax is the maximum possible initial reaction velocity. So the substrate concentration that produces half the maximum velocity is known as the Michaelis constant. Now, what is the meaning of Km or Maclaurin's constant? So, it has been determined for many of the commonly used enzymes. The size of Km will tell us several things about a particular enzyme. So, a small Km will basically indicate that the enzyme requires only a small amount of substrate to become saturated. Hence, the maximum velocity is reached at relatively low substrate concentrations. However, a large Km will indicate that the need for high substrate concentrations to achieve maximum reaction velocity. The substrate with the lowest Km upon which the enzyme acts as a catalyst is frequently assumed to be the enzyme's natural substrate, though this is not true for all enzymes. So, a Km of 10 to the power minus 7 molar indicates that the substrate has a greater affinity for the enzyme than if the Km is 10 to the power minus 5 molar. So, what is the significance of Michaelis maintained Michaelis constant Km? It's an enzyme kinetic constant. It indicates substrate concentration required for the enzyme to work efficiently. Low Km indicates high affinity of enzyme towards substrate and vice versa. Hence, Km affinity. Exokinase and glucokinase, you can see Km of exokinase is low, whereas Km of glucokinase is high. Km is required when enzymes are used as drugs, and use of enzymes in immunodiagnostics require Km of the enzyme. So the catalytic constant Kcat. Now at high substrate concentration, the overall velocity of the reaction is Vmax and the rate is determined by the enzyme concentration. So the rate constant observed under these conditions is called the catalytic constant Kcat, which is defined as Vmax Kcat E total. Now the Kcat indicates the maximum number of substrate molecules converted to product each second by each active site. This is called turnover number. So the catalytic constant measures how fast a given enzyme can catalyze a specific reaction. And the unit for Kcat is second inverse. Now if we make a double reciprocal plot, that is the line Weaver plot, which shows it basically inverts is a double reciprocal of both the sides. So it will give 1 by V naught equal to Km by V max x plus 1 by V max. Okay, so this is a line Weaver double reciprocal plot. Now for kinetics of enzymes, we know enzymes can follow zero order kinetics when substrate concentrations are high, and zero order means that there is no increase in the rate of the reaction when more substrate is added. Okay. So if you see this is the equation basically that has given rise to the Maclaurin equation. So when the substrate concentration becomes large enough to force equilibrium to form completely all enzyme substrate complex, the second step in the reaction becomes rate limiting because no more ES can be made and the enzyme substrate complex is at its maximum value. So ES is the difference between the rates of enzyme substrate formation minus the rates of its disappearance. So for assumption of equilibrium, we know that the K minus 1 is much greater than K2. The formation of product is so much slower than the formation of AS complex. We can assume Ks is ES by ES. So the K is the dissociation constant of the EX complex. Assumption of the steady state. Now transient phase where in the course of a reaction the concentration of ES does not change. So this is basically the derivation of the Maclaurin constant. You can find it in any biochemistry book as well. So these steady state assumptions and all of these give rise to the Maclaurin equation. And this is the Maclaurin curve. A K with substrate concentration where V naught equals 1/2 of V max. The Km or the Maclaurin constant varies among different enzymes. Okay, so as if you can see, as Ks decreases, the affinity for the substrate increases. So the Km can be a measure for substrate affinity. So double reciprocal plot is very important because the line Weaver plot actually helps us in analyzing various kind of kinetic data. So basically the line plot, the intercepts, the extrapolation, everything will give us some kind of information. Now Kcat is basically the turnover number which shows how many reactions an enzyme can catalyze per second. and Kcat by Km is a measure of catalytic efficiency. Now, what is catalytic perfection? So, when every substrate, when K2 is greater than much greater than K minus 1, or the ratio is this ratio is maximum, or when every substrate that hits the enzyme causes a reaction to take place, that is catalytic perfection. So, diffusion control limit diffusion rate of a substrate is in the range of 10 to the power 8 to the power 9 per mole per second. So an enzyme lowers the transition state, so there is no activation energy and the catalyzed rate is as fast as molecules collide. So there are some important things to remember about the concentration of substrate. Okay, so at low substrate concentration, not all enzymes are saturated, so the rate of reaction will increase. At higher substrate concentration, all enzymes get saturated with substrates, and any more increases substrate concentration will result in no increase in the rate of the reaction. For the Michaelis-Menten equation, this equation describes the dependence of reaction velocity on substrate concentration. Michaelis and Menten propose that in any enzymatic reaction, the enzyme combines with substrate to form an enzyme-substrate complex. ES then breaks down either to form enzyme and substrate again, or to enzyme and product. The Michaelis and Menten equation describes how reaction velocity will vary with substrate concentration as follows over here. So from Michaelis constant, from the above equation, when substrate concentration is equal to Km, thus Km can be defined as substrate concentration that produces half maximum velocity. Now, what are the important conclusions about Michaelis-Menten kinetics? So, substrates are usually present in physiological fluids in amounts nearly equal to Km values. So, Km is a constant characteristic of an enzyme and its particular substrate, and Km reflects the affinity of the enzyme for the substrate. The smaller the Km value, the more active the enzyme. So, small or low Km reflects the high affinity of the enzyme for substrate, that is, low concentration of substrate is needed to half saturate the enzyme, and large or high Km reflects a low affinity to the enzyme for substrate, that is, high concentration of substrate is needed to half saturate the enzyme. Then we talk about the line-Weaver-Burk plot, which we have over here, double reciprocal plot. We talked about the enzyme inhibitors. These substances basically that can diminish the velocity of enzymatic reactions. We've already talked about that. So many questions regarding this. Okay. So we can stop here for today. If you have any questions, you can ask me. Depend much on the question that we discussed today. Any okay? So if there are no questions, we can stop the session today, and we'll meet again next week, Monday. Thank you.